In this chapter, we're going to start looking towards DNA, structure, and organization. So this is part of a three-part series about DNA modeling completely. So first, we have to learn about DNA structure and organization, see what it's like physically, and then we'll be able to use our knowledge of physics and different tools to be able to model and examine the behavior of DNA in different ways. So we're starting off this with just the DNA helical structure, how it can be unwound and wound. And then in chapter 10, we're going to see how we can model just one strand of DNA, which is called the random walk model, or a worm-like chain model. Then from there, we can try to see if there are different ways of looking at DNA as well, from its charge distribution to its electrostatic interaction, to wrap around histones to form chromosomes. So we are starting a new topic on DNA. So there's not a lot for us to bring forward from our knowledge of proteins and enzymes, but we are going to go back to the beginning of the class and discuss unity at the molecular level again, and just think back to what that meant. So what this was that whenever you're looking at a macromolecule, you can simplify it to its basic building blocks. For proteins, it was amino acids, and they acted as the monomers, and we were able to model amino acids in different ways, whether it was polar or nonpolar. Now, when we're looking at DNA, its building block is a nucleotide, and those nucleotides will also have different properties that we can take advantage of for modeling purposes. So our unity is now to look at nucleotides for nucleic acids to see how we can model their behavior in different ways. We did take a look at hydrogen bonding and protein structure in the past, so that's going to come into play with this lecture as well, because in order for those monomers of nucleotides to bond or bind together or be connected together in some way, there has to be some sort of interaction. So that would be discussed through a hydrogen bonding network to see which types of nucleotides can connect well together. So for the main lecture, we are going to look at the structure and organization of DNA. We're going to take a look at how DNA is packed with histones, and then we're going to go through a high-level summary of different ways to model DNA in preparation for future chapters. Okay, so let's first go through DNA and its organization. So first of all, we all know that DNA is made of nucleotides. So it's made of 4 nucleotides, which are building blocks. Every single nucleotide here has a sugar. So that's a picture of a nucleotide. There's a sugar. There is a phosphate that's linked to a base. So sugar plus phosphate to create a sugar phosphate, plus some sort of base to create a nucleotide. It's this base that gives each nucleotide its unique feature and a unique name. Because there are 4 bases, it means that there are only 4 different types of nucleotides. So we just simply call them 4 bases A, C, G, and T. They are covalently linked together, and the arrows point in the direction that they run, and they are anti-parallel to one another, forming a double helical structure. So that's the high-level overview of what DNA is made of. In terms of these different bases, though, we talked about the hydrogen bonding network before. You'll see these little dashes in between these base pairs, or these bases. This is one set of base pairs, and they are going to be connected to each other through hydrogen bonds. So that's the next slide. We see here that the shapes and the chemical structures of the bases, so these are each of the bases. There's adenine, guanine, cytosine, and thymine. They have different shapes and they have different projections. And so because of that, it'll allow different quantities of hydrogen bonds to form between these bases. For A and T, they only have 2 hydrogen bonding opportunities, and so therefore they must always bind together. So you create an AT base pair. You then have cytosine and guanine, each being capable of 3 hydrogen bonds, so they are always paired up together as well. You end up with A's and T's and C's and G's always connecting with each other to form a 2 or 3 hydrogen bonded connection. So looking at the image on the right, we have now a 3D structure of what these bonds look like in relation to one another. 4 of these base pairs are shown. So you have CG, AT, CG, and CG. What's important to note is that there is a sugar phosphate backbone that runs alongside each base pair. And to connect these sugar phosphates together, there's what's called a phosphodiester bond. So that's an important term to remember. That phosphodiester bond is going to link nucleotides together and gives each strand what we call a chemical polarity. So the 2 ends are actually chemically different. So we call one the 5' end and the other one the 3' end. The 3' end always carries an OH or a hydroxyl group on the sugar ring, and then the 5' end is going to carry a free phosphate group that's attached to the 5' position on the sugar ring. So what does DNA help with now that we know its overall structure and how it comes together? So if we take a look at this picture, we see that there's a DNA double helix and a few regions that are highlighted in different colors. So different regions of DNA are going to contain different genes that code for different proteins or functions. So as our body reads the DNA double helix, it'll hit gene A, which will then be transcribed into RNA A, let's just say for gene A. And that RNA is then going to be read to create protein A. So different regions of your DNA gives you a transcript to read off of to build different proteins. So as you can see, if any part of that process goes wrong, you end up with an incorrect protein or a lack of a protein being created, which causes potential disease. So apart from that, we also have to understand how DNA can be organized. So we've looked at DNA by itself as its helical structure and the hydrogen bonds that form within. We've taken a look at DNA as a whole and how it's being read. We'll get into the details of that later on. Now, once we have the DNA strand, how can it further condense or further pack itself so that it's one piece? Because the strand that we saw before is just a small snippet, there's a bunch of different genes that a human or any living creature can have. So how can this huge genetic code be stored in DNA? So one of the things that's really important to understand is that DNA has to be highly condensed so that all this genetic material can be stored in a small 3D space. So that's why we have to take a look at chromosomes. In a eukaryotic cell, basically very long double-stranded DNA chains are packaged into structures called chromosomes, and they contain a long string of genes that can be read. And so these chromosomes can be unwound at any point in time to create those proteins. So there is a detailed folding process to get a chromosome, but first let's take a look at what chromosomes are and then take a look at the folding process, because that will come into play in future chapters. And just so you know, there are so many different ways of modeling DNA that this lecture is simply just an overview of all the different ways we can look at DNA from its chromosomes down to its individual hydrogen bonded network. So human chromosomes, in picture A, we basically have an image taken after a cell was broken up or lysed. In B, they were organized artificially. So basically they took pieces of this and organized them by color and by shape and size. And they were paired together for easy identification. So based on this, after you lyse a cell, you have all these chromosomes, and you end up with a perfect pairing of 23 chromosomes for the human genetic code. But if we take a look at one chromosome over here, it's more than what we just see. If you start to unwind it, you'll eventually be able to go back to your DNA double helix. And so we're going to start here with the DNA double helix and slowly wind it so that it ends up being a chromosome. So how is DNA packed? So it starts with the DNA double helix, which is formed by your sugar phosphate backbone, connected by phosphodiester bonds, each connected to, each of those sugar phosphates is connected to a nucleotide or a nucleotide base. And therefore, you end up with a bunch of base pairs that work its way around that double helix. So the DNA itself is first going to wrap around a structure called a histone. And so this is where we start looking at electrostatic attraction. And that's what one of the future chapters will be based on, how chromosomes, or how DNA is going to be attracted to histones and how it wants to wrap around histones. So as you can, if you remember from previous lectures or from other courses, DNA is negatively charged. If it wants to wrap itself around a histone, these yellow things over here, yellow discs, if it wants to wrap around a histone, then what's going to happen is it must be attracted to it. So the histone most likely is positively charged, which we'll discuss later, and the DNA is going to be attracted to it and want to wind itself around it. So you have a bunch of these histones. You have DNA winding itself along many of these histones, creating a bead, basically a beads on a string formation. And this is then going to wind or condense into something like this. So it's going to wind itself. And so this creates a chromatin fiber of packed nucleosomes. So each one of these beads is called a nucleosome. Once DNA wraps around a histone to create a nucleosome, all the nucleosomes are going to want to wrap around itself to create a packed nucleosome environment. Once that's done, it's going to continue to wrap around and condense to create a chromatin fiber that's folded into loops. So you basically have the histones going up and down like this, and being condensed and packed along this orange region over here. And so if you take a look at the entire strip of DNA and have it all condensed through these histones, you end up with an entire chromosome with it being bound in the center by a centromere. So each DNA molecule actually is able to be packed into a mitotic chromosome that is 10,000 times shorter than its original length. So that's pretty cool. So this is something that we'll focus on in 2 chapters, I believe, where we talk about how this structure is possible physically and entropically. So taking a look at one of these beads, this is the first electron microscopy picture that was taken of a nucleosome. This is in B, where you have the beads on a string. That's exactly where that term came from. Chromatin was isolated from nucleus in the interphase of mitosis, and it appeared as 30 nanometers thick threads along this image. And when these were decondensed experimentally, nucleosomes were shown. So it's pretty cool. So if we were to look at the nucleosome just by itself graphically, then we would use this as a histone, the circular or the cylinder, and it can be split up into 8 pieces because 8 pieces make up a histone octamer. So once we have this octamer here, the DNA is going to wrap itself around the octamer X number of times, and that is one of the pictures that we'll focus on in the future to start our analysis of DNA as a giant charge. So back to what we were discussing at the beginning, how can we model DNA? We can model DNA in many different ways, and we are going to go through a few of these ways in subsequent chapters. So we're going to be looking at the sequence of DNA. That means we have information provided to us from the sequence so that we know exactly what kind of hydrogen bonds are taking place, how many of them, and what kind of structure we have. In the next one, we have a binding site modeling system. Basically here, we have protein binding sites, which allow us to study the interactions of DNA with other molecules. This is the one that we'll be focusing on the most, charged rod. So in the charged rod model, we model DNA as a giant charge, a giant negative charge, because it has polarity, and see how it interacts with other molecules like histones. These two are quite well tied together. We can also model DNA as an elastic rod through a worm-like chain model. And through the worm-like chain model, which is based off of a random walk model, we can also assume that each of those persistent lengths is a spring. So we can look at molecular springs and random walk models depending on the size of DNA we have to see if it can tell us some information about how much DNA can bend, as well as how much it can stretch. The main reason for this is because when it comes to DNA, and the central dogma of molecular biology, we have to be able to read DNA and create proteins. So that means if the DNA is condensed, there has to be a way to unwind it and stretch it so that some sort of protein can come in and read it. So being able to model DNA's elasticity and the way it untwists or twists again, that is really useful, and that's the focus of the next chapter. So that was a pretty broad overview of DNA and what we're going to be focusing on in the next few chapters, including this one. What we want to do now is really get into the details of DNA structure and how it can stabilize itself. So being able to analyze the structure and properties of DNA, the energetics, and how stiff it actually is, that is important to know once we move on towards being able to model DNA when it's denatured, and so on. So first of all, we have another picture over here. Basically here we have nucleic acids that can be presented in different ways, proteins that can be presented in different ways. So I'm going to start off with proteins. With proteins, we were able to model them using an alphabet system. So that's basically like a sequence. There were basically a bunch of amino acids that we could have used, which refer to the alphabet. We only have a certain number of letters we can choose from, and there were 13 amino acids we could choose from. With nucleic acids, there are 4 different nucleotides we can choose from because there are 4 different bases that can exist. So that's the limitation that we work with if we use the sequence form. Now, the next step of proteins we looked at, we said that these amino acids from that sequence, after hydrogen bonds are introduced and they form some sort of structure, they form an alpha helix or a beta strand. With nucleic acids, the next level from alphabets towards, or basically to their secondary structures, we create codons. So a set of 3 base pairs that provide a sequence, and these sequences actually help determine which amino acid to form. So the next level from the monomers or the sequence would be your codon, which is a part of your DNA double helix. And if we go to the last level, when we're forming sentences from those words, if you have an alpha helix and a beta strand for your protein, eventually you end up with a tertiary or a quaternary structure, which allows for functionality to take place, and you end up with a complete protein with different pieces of alpha helices and beta strands. When we talk about nucleic acids, that means we've completed the entire DNA double helix, and we have an entire gene. So that is the end product of each polymeric chain that we're trying to create. So in terms of DNA versus proteins, some of the key differences are DNA is going to provide instructions to create proteins, and DNA is going to help translate between polymer languages. DNA is considered a mechanism for reading and writing. So that's why a lot of the analogies have to do with language. So genes are going to know how to make the proteins, and proteins are the little robots that can carry out the functions. Some DNA electrostatics to start the conversation. DNA is negatively charged. So if you are to model DNA electrostatically, you have to use a negative charge for it. It's actually approximately negative E over 1.7 angstroms. So that's the exact negative charge that it has. DNA is packaged with what we call multivalent cations. So when you're packaging DNA and condensing it, you need 2 or 3 positive charges in order for it to be stable. So it then can form these toroidal bundles with histones. If you have a salty solution with a 3+ charge, then DNA strands may be attracted to each other. That's just something interesting to know about. And if we're taking a look at DNA in other organisms, they have a very similar packing structure, including viruses as well. So what about eukaryotic DNA? So when we take a look at eukaryotic DNA, it uses electrostatics to pack itself. In red, we have our DNA, and in yellow, we have our histone octamer. One thing I didn't show before is the histone tails. There's actually projections of the histone so that there are more areas of positive charge that can attract the negatively charged DNA. So there's actually these negative tails that stick out, and with all of this, there are approximately 200 positive charges on one histone that will attract the DNA to bind around it. So how does the DNA wrap around a histone? Both the DNA and the histones are almost all neutral. So even though there is a negative and a positive charge, if you take a look at the whole thing, they're primarily neutral. So the direct electrostatic interaction is very weak. When we do the modeling, we have to take that into consideration because we cannot just assume a tiny positive charge is very positive. So we have to come up with another explanation to allow this to make sense. So when they are close, when the DNA and the histones are close to each other, their backbone charges are going to start to neutralize each other. The reason why is because the main driving effect of all this is the entropic gain of released counter ions and not energy. So I won't discuss this in detail until we reach one of the last chapters of DNA, when we're discussing this in a lot more detail. But the key thing here is that electrostatics will allow DNA to pack itself, but it's not going to help stabilize it significantly. It's the entropic, there's an entropic reason for why it stays like this, and that's something that we'll have to discuss. So everything that we take a look at cannot just be explained by one thing. It's actually multiple different components coming together to offer a full explanation for DNA stability. So that's one way of modeling DNA. Another way of modeling DNA is like a garden hose, or basically like a random string. If we take a look at DNA, if we take a look at a short section, it's actually pretty stiff because it's a double helix and it's hydrogen bonded with those base pairs. So it's almost considered linear with minimal curvature if you're looking at a short piece of DNA. So we consider this part to be locally stiff. But if you have a really long strand of DNA, then that's a totally different story. Because if you piece a whole bunch of different locally stiff components together, you can end up with a lot of flexibility. So at large length scales, above or equal to 500 angstrom, you can get a much wider conformational space to increase entropy. So this resists stretching or confinement, and we now have a much better explanation and definition for what we call persistent length. So this is what we call persistent length. That is the length of DNA that is considered linear. We have taken a look at persistent length before, and we are going to take a look at it in a lot more detail now. So The persistent length is 500 angstrom because we call it locally stiff. Anything past that, we can consider it to be a coil-like state. So when we take a look at the DNA in a short section, we consider it as a twisted ladder. A ladder is very stiff. We have a persistent length that's much longer than this actually, and it still remains fairly stiff, but it has to maintain that stiffness because it has to maintain a twisted ladder shape. So because of the different types of hydrogen bonds available for the base pairing, that is the cause for that stiffness. So if there's any questions that pop up that encourage you to explain why the persistent length is why there is a persistent length and why it is 500 angstrom, it's because of the base pairing and the hydrogen bonding between the base pairs that allows for that stiffness to be prolonged to 500 angstroms. So here's just another visualization of the helical structure of DNA. You have a ball and stick model, and you have the space filling model, which is a lot more difficult to look at if you're just looking for the hydrogen bonds. Now, because we have taken a look at how alpha helices before, remember in our discussions with the Ramachandran plot, there's different versions that exist depending on how something twists. For protein alpha helices. Same thing for DNA helices. There's actually 3 different forms. There's the A, B, and the Z form. The A form and the B form, they can be separated. Actually, all 3 versions can be separated based on how many phosphates there are per turn. So in order to determine which type of helix you have, you just have to count how many phosphates exist in the backbone for one turn. For the A form, there's 11 phosphates. For the B form, there's 10 phosphates. And for the Z form, there are 12 phosphates. So 11, 10, and 12. Easy way to remember this in the order would be 10, 11, 12, or BAZ. So moving forward, we want to be able to model DNA graphically. So one of the best ways of doing this is to simplify things. When we take a look at hydrogen bonding between base pairs, we notice a few things with the base pairs as well as the hydrogen bonds. So let's take a look at the base pairs first. No matter what, you always have one base pair being formed between a base with 2 rings and a base with 1 ring. The base with 2 rings is called a purine, and the base with 1 ring is called a pyrimidine. So basically, it's always 1 purine and 1 pyrimidine that are bonded together. So we're going to create one shape that represents purines and another shape that represents this. So we'll have A like this, and A and G in this shape, and T and C in this shape. And we're going to create a notch in here and an extension here, just so that we know that A and T have to connect together. And we're going to color code them because A and T must only connect together because they form 2 hydrogen bonds, and G and C must connect together because they form 3 hydrogen bonds. So that is a simplified way of drawing these bases so that they connect to each other, and we'll be able to tell which base pair we're looking at based on color or shape. So we will discuss that in a future chapter. When we take a look at DNA bending, we also have to worry about which way it goes. So which way does DNA tend to bend? Once again, we're able to calculate the energy penalty. If we take a look at short lengths, the free energy is going to win and DNA is going to remain straight because there's nothing else urging it to bend in a certain way to increase entropy. At a large length, though, if you increase the length of this, then the entropic effects will start to take place. It's more entropically favorable if you have a really long strand and it starts to want to turn into a coil-like structure. So at large length scales, entropy is going to win over the persistent length, and DNA is going to start to coil up to increase the entropy of the entire chain itself. Otherwise, if it is under a certain length, because it is fairly rigid, it will want to remain pretty straight to conserve its energy. So this allows us to link energetics and stiffness together. When we take a look at base pairing and stacking, those energetics can be translated into chain stiffness as well as persistence length, LP. And one of the things that we are going to take a look at in the subsequent chapter is the worm-like chain model. We use this to model such a behavior because we're going to model this entire wormy chain using the WLC. Once we have that model available to us, then we'll be able to take a look at things like DNA denaturation. So that's if we have DNA thermal denaturation, it involves heating up your DNA so that it becomes denatured and it completely unwinds into single-stranded coils. There's a melting temperature, Tm, that's associated with this. That's related to DNA stability, depending on what you have. Not just this, but once we have that worm-like chain model, we can also introduce what's called the zipper model. That's what happens when DNA denatures from one end. So it opens up in a small pocket and it completely unzips. And that is something that we can also use the cascade effect for, because when you unwind one, then it starts a cascade effect and continuously unwinds the subsequent base pairs. So this is the bubble that I was talking about. When we are denaturing DNA using the zipper model, we can always start at a specific location called local denaturation. If we start here, we can unzip it. The bubble formation is catalyzed by DNA untwisting. So there has to be some sort of energy input to allow the untwisting to occur. Once it occurs, there's a cascade effect, which unwinds the DNA in this bubble region. And this area effectively reduces the melting temperature to allow for DNA to be unwound a little bit easier. So what did we learn today? We just learned a really high-level overview of the different types of ways we can look at DNA in preparation for our upcoming chapter, which is the worm-like chain model for DNA. So from this chapter, you should know what DNA is made of, explain its structure and interactions, and how it folds around histones, and ultimately into a chromosome. And of course, you should understand that we can model DNA in different ways to get different information that we're interested in. So more details in subsequent chapters. This is just a brief overview of what's to come.